Viruses are the smallest infectious agents known to men. They inhabit every ecosystem and infect every living organism, whether plant or animal. Although there are probably over 1 million types in existence, about 5,000 has been described in detail. Some threaten our health, while others threaten our food production systems. Some have the ability to remain dormant in a host for many years and only causing a full-blown disease when the host immune system has been compromised. Many may cause nothing more serious than a common cold, while some are implicated in being cancer triggers. Viruses mutate frequently, and once in a while a new mutant strain emerges that can cause serious outbreaks that may reach pandemic proportions.
Viruses are potentially very deadly to mankind. For the 18 months of the Spanish flu outbreak from the middle of 1918, 500 million people were infected, of which an estimated 50 million died.
Biovalence is a new startup company formed by a group of interested investors specifically to bring Defensia's drug discovery platform to the next level. Over the next few years, Biovalence will be investing millions of dollars to build a US FDA standard CGMP clinical batch production facility as well as a GMP facility for animal drugs compliant to the Department of Veterinary Services Malaysia. Biovalence will also support further preclinical research and to bring Defensia's technology to US FDA Phase 1 completion.
VER.1 may be the first of a whole class of new drugs. It is an oral delivery broad-spectrum antiviral. It is a protein drug. It is made using genetically engineered bacteria that produces this protein.
Instead of using painstaking conventional drug discovery methods that sometimes is more like finding a needle in a haystack, we opted for intelligent design. The fixing up on sets of antiviral genes from different genomes into fusion proteins that target multiple sites of the virus cycle. VER.1 could be amongst the world's first few multifunctional antiviral drugs that show broad-spectrum applications.
By fixing genes together using modern molecular biology, an entire new pipeline of bacterially expressed antiviral drugs are now being made in this lab. VER.1 has already shown activities against 10 different types of viruses in either in vivo or in vitro trials. Our first human indications are likely to be for oral and genital herpes and dengue fever.
Antivirals basically will depend also on which viruses, whether the vaccines are available and how good the vaccines are. And usually vaccines for dangerous organisms, you have to use a killed vaccine or a sub-unit vaccine, which are all really suboptimal. Therefore, it's better for us to actually target the virus directly with a specific antiviral or even a broad-spectrum antiviral for that matter. The viral transmission route as well. For other viruses, symptomatically, certainly the broad-spectrum antiviral will be very, very useful. And I think for viruses like cytomegalovirus, other latent viruses like HSV, BK viruses, when people are undergoing transplants, they are immunocompromised, and hence these latent viruses take advantage. And in these instances also, broad-spectrum antiviral will be very useful.
Cats and dogs are very important for humans as their pets. We have been looking at 3 important viruses: feline immunodeficiency virus, FIV, feline leukemia virus, FELV, and canine parvovirus 2 in puppies.
While we are gearing up for human trials, we have begun providing VER.1 as an experimental drug for cats and dogs.
Brownie was rescued and found to be suffering from parvovirus, and he was in really bad state, dying, and the vet said his chances are almost nil.
About a year ago in December, Ao got into a small accident and he got a small cut here in his hip and it got infected. And I took him to the vet and he had to have it stitched, but he got hepatitis. And we knew he was FIV positive and therefore he wasn't recovering properly. But by January, he'd gone from about 4 or 5 kilos to 3 kilos. And then he went to 2.8.
We had a cat with very bad secondary infection, ulcers all over the body and head, which did not respond at all to conventional treatment. We thought all that we could afford to provide him at that time was just palliative treatment.
1 year ago, I had already heard about VER.1, but I didn't know it was effective for canine parvovirus. I thought it was only FIV and FELV for cats. So this friend recommended that I try VER.1, so I got in touch with Mr. Huan and Yanti. We got the VER.1, we gave it to Brownie, and he recovered miraculously. I hope VER.1 will be marketed so that we can reach out to pet owners.
I really thought time was coming where I'd have to put him down because he was beginning to look unhappy. Then I remembered that Sabrina Yeep from Friends Furry Farm had told me about this magical goop that she'd been using on some of her FIV cats. And I called her and asked how it was going, and she said they'd all recovered. So she gave me the number for Benny Tan in Puchong. And I took Ao there and I got him signed up on the trials. And within about 5 days, he started eating properly. Yeah, 3 or 4 times a day. And he was waking me up in the middle of the night to tell me he was starving, he wanted food. And he started putting on weight. Yesterday, he was 4.5 kilos.
We got the opportunity to do the first trial on VER.1 on cat. This is where it all started about 2 years ago. I was introduced to Juan. Then he passed to me this VER.1 to be used on Orange the cat. We started to see improvement on his condition basically about 10 to 14 days after the initialization of the therapy. What we first noted on him was that the ulcers started to become less angry, bleeding much lesser, and we get a complete recovery from his condition about three months of continuous oral medication using VER.1 and the conventional treatment. Yeah. And ever since that, I've tried out this VER.1 on other viral cases as well, namely canine parvovirus, feline parvovirus, feline leukemia viruses, and all these 3 other viruses. We have got very encouraging results as well. The balance has changed. It seems like we have got the upper hand. Most of the cases, if we get it early, detect it early, we are able to save the lives of the puppies.
VER.1 was completely developed and validated by Malaysian scientists.
There's growing evidence that viruses trigger off cancers. Some researchers believe 20 to 40% of cancers might have a viral trigger. We have begun experimenting on one case of a pontine glioblastoma, a brainstem cancer, where we have seen the cancer regress from a 5 centimeter diameter to 2.5 centimeter diameter without any side effects in a 13-year-old boy. We are still closely monitoring this situation. He was initially given only 3 months to live, but it has been almost a year since then.
Another US company, also developing broad-spectrum antivirals, was recently funded by the US NIH to the sum of $45 million. Such is the value of similar technologies.
The world antiviral market is now worth 28 billion US every year. We have the option for an unlimited pipeline of chimeric antimicrobial peptide drugs.
Our dream is to put Malaysia on the map, and our hope is that VER.1 can save companion animals and later on humans.
We know that drug development is a long-term process. We, at Biovalence, are in it for the long haul.
I am Biovalence.
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 We are Biovalence.